Hey everyone. Today we are talking about our research project titled of Investigating Physiological and Environmental Impacts on Early Onset Alzheimer's. My name is Anamika and here are my other teammates Sarah, Michelle and Anthony. We are group 3. Basically we are exploring how factors like our health and environment can influence the onset of Alzheimer's at a young age. It's very important step and we are excited to share what we have found. Let's jump in. Dementia. It's a significant global health concern. It's ranked as a seventh leading cause of death. Surprisingly, its impact about 50 million people worldwide, creating huge pressure on families and society. Experts are saying that number is will be triple by the year of 2050. Now, when we talk about dementia, we are actually referring several different types, including Alzheimer's disease, frontotemporal dementia, and vascular dementia. Among these, Alzheimer's disease take the spotlight as this is the most prevalent and accounting for 50 to 70% of diagnosed cases. But here's the interesting part. While we often associated dementia with older adults, young adults can also experience cognitive declines, especially with early onset Alzheimer's disease. This presents a unique and significant challenge that researchers are still trying to fully understand. Trying to find solution for early onset Alzheimer's is not easy, but it is incredibly important for individuals, their families, and society at large. When we talk about dementia, we are referring to disruption in brain function that affect how nerve cells communicate with each other. This can result in cognitive impairment and changes in behavior. Now, early onset dementia is a bit different. When these symptoms pop up before the age of 65, which can present some unique challenge for both individual experience it and their families. And what really interesting is Alzheimer's disease is the most common type of early onset dementia. We have probably heard it before. It is condition where a significant number of cases occur before the age of 65 and they are linked with Alzheimer's. But the twist is genetic factors only play a small role in early onset Alzheimer's. A specific gene mutation like the APP and PSEN1 and 2, they are responsible for just a fraction of cases. Instead, environmental factors become important. Factors where we live, what we eat, and how much education we have had can all impact our risk of developing early onset dementia. So, why does all of this matter? Well, understanding these factors is crucial for developing effective prevention and treatment strategies. By digging into the root causes of early onset dementia, and with this knowledge, we hope to make life easier for those affected and their families. Now, let's talk about our research question driving our research project on Alzheimer's disease. First up, we are digging into clinical data to figure out what's different about young people who develop Alzheimer's compared to those who get it later in life. We are looking at things like gene, body function, and all sorts of factors to see what might causing Alzheimer's to show up earlier. And why does all this matters, like its importance? Well, focusing on early onset Alzheimer's is a super important because it helps us understand what's going on the younger brain. Plus, by exploring a bunch of different factors, we can get a clear picture of what might be causing Alzheimer's in the first place. Ultimately, our research isn't just about asking questions. It's about finding answers that could lead to better way to prevent the treatment of Alzheimer's. So, by unraveling these mysteries, we are hoping to make a real difference in the lives of those affected by this disease.
Hello, everyone. My name is Sarah Avali, and I'm here to go over the methods with you. First, we started by choosing a database. We chose the NACC database, the National Alzheimer's Coordinating Center. They have a lot of clinical diagnostics of Alzheimer's patients, including clinical assessments, demographic data, autopsy reports, MRI, and CSF data. I have the website here linked in case any of you are interested. We did get the data by request, so we had to request it, wait a few days for a response, and then they were able to send us the data that we needed. It was a very large data set, approximately 48,600 plus participants, 180,000 plus clinical assessments, 7,565 neuropathology exams, approximately 33 active sites across 26 states, and they're currently expanding their participant diversity. That's super important when you're trying to create a very universal data set that covers all, genders, nationality, all of those demographics. Here we have a map of the sites. Now, these are states that are participating and then also specific locations here in yellow. So for our methods, we started with data cleaning. We used the pandas and NumPy packages. We separated those out into categories. We combined them by age at first visit, and sex to both CSF and MRI data sets. And then we did that by ID. We replaced all null values to non. And then we further those categories. I wanted to explain a little bit further into how we did that. This data set was really, really large, so we had to break it down into smaller categories. So we broke it down into sets of 10 variables each, and this was with it's called the UDS data set, which was the survey data. It was full of qualitative data. And so we separated those out, and then we performed scatter matrices using Seaborn on both the UDS data set, which was the survey, and then the MRI and CSF data. Then we performed linear regression on the quantitative data using scikit-learn. The quantitative data in particular was the CSF and MRI data, and to see those linear regression plots, which we'll go into further in a little bit. We then did Pearson's correlation analysis, or PCA plots, on the qualitative data using Plotly. And this was the UDS in particular, which was all of that survey data, had a ton of demographics, a lot of really good information about the patients in particular with Alzheimer's. Then we did box plots on those qualitative data that showed correlation on those PCA plots. And then we used matplotlib to pyplot to plot those. And then we'll go into that further. My next cohort will go into that in further detail. Thank you for your time.
Beginning with about 10,000 variables, we culled string data types and culled data types that have no entries. This left us with about 1,000 variables. From that 1,000, we chose the most relevant demographic categorical survey data, and that was about 80 variables. We included all quantitative variables, which totaled about 30 variables, mostly from neuropathology autopsy records. From these variables, we produced a collection of scatter matrices that each includes age and sex and about 5 variables for each matrix. Producing about 50 matrices, for each of these scatter matrices, we analyzed the top row, which indicates the relationship of age for any given variable. If the plot output was categorical and visually indicated to share an age difference, as seen here, we would assign it as such. Conversely, if it produced a scatter plot and visually indicated even slight correlation, we would include this and assign it separately as quantitative. These two groups of data types were then separated into different data frames and analyzed accordingly. Once visually indicated significant variables have been identified and grouped as categorical or quantitative, the categorical data was analyzed through principal component analysis to derive the variables with the highest influence on the principal component. The top three were identified as the lumbar puncture year, which is a sample of cerebrospinal fluid from the lower back that operates as fluid flowing around the spinal cord and the brain, which clears waste products. Levels of certain proteins in the cerebrospinal fluid reflect brain physiology, as well as T-tau assay method, which explains the method of quantitative analysis of metabolic hormones and cytokines. The enzyme-linked immunoabsorbent assay, or ELISA method, is considered the standard for such a test, and the Luminix multiplex bead array assay was developed as an alternative to ELISA. And finally, education level, which represents the level of education that any given patient was able to accomplish. The results of these box plots seem to show little to no difference, with the strongest being lumbar puncture year. This, however, is not the limitations of the categorical data here. As we can see, box plot generation does reveal other variables that have age-related differences. We can observe age-related differences in which inherited Alzheimer's disease mutations is causing a patient's disorder. This is labeled as NACCAM. There's also the total number of medication use per patient, labeled as NACCAMD, and posture status, labeled POSTURE, which is a general scale of 0 to 4 that measures a patient's flexibility and spine health. Regression analysis was performed on all remaining quantitative variables, with the strongest correlations by at least 1 order of magnitude. Plotting these regressions revealed a slight negative correlation on the right pars orbitalis mean cortical thickness, RPAR-ORBM, a negative correlation in right pars triangularis gray matter volume, RPAR-TRI, a negative correlation in left paracalrine mean cortical thickness, LPER-CALM. Finally, a positive correlation in segmented total third ventricle volume, labeled here as DHIR-VENT.
Hi everyone, my name is Michelle and I will be presenting information on future implications, conclusions, and our references. Our research contributes to the broader understanding of early onset Alzheimer's disease research, offering insights that extend beyond our study. The identification of specific biomarkers and environmental factors not only enhances our understanding of the disease, but also holds promise for future research endeavors. By building on this foundation, researchers can further investigate the relationships between influencing factors and develop more targeted approaches to diagnosis, treatment, and prevention. Our findings emphasize the importance of personalized medicine in the management of early onset Alzheimer's disease. Recognizing the unique genetic and environmental profiles of individuals affected opens avenues for tailored treatment strategies, potentially improving patient outcomes and quality of life. Furthermore, our research has implications for public health initiatives and policymaking. By investigating the epidemiology and risk factors associated with this disease, we provide valuable data to inform healthcare policies, resource allocation efforts, and community-based interventions aimed at mitigating the impacts on individuals, families, and society. Our findings ultimately contribute to a growing body of knowledge that will shape the future of early onset Alzheimer's disease research and clinical practice. Such insights will allow healthcare to advance towards more effective prevention, diagnosis, and management strategies, ultimately striving towards improved outcomes for individuals affected by this challenging condition. Our analysis revealed specific variables such as changes in cortical thickness, gray matter volume, and ventricular volume observed in MRI scans that demonstrate correlations with both age and disease progression. These findings suggest potential biomarkers and environmental factors that may contribute to the pathogenesis of early onset Alzheimer's disease. Utilizing advanced analytical methods such as multivariate analysis, regression modeling, and visualization tools such as scatter matrices and principal component analysis, we've identified specific biomarkers and environmental factors that are strongly implicated in the pathogenesis of early onset Alzheimer's disease. Our study emphasizes the importance of considering both genetic and environmental factors in shaping disease risk and progression. With these factors in mind, we can better assess disease risk, facilitate early detection, and implement preventative measures, ultimately working towards improving the lives of those affected by this devastating condition. Our research represents a significant advancement in the fight against early onset Alzheimer's disease, but it's important to recognize that our work is just the beginning. Further research and validation are essential to confirm and build upon our findings. By continuing to investigate the environmental and physiological factors surrounding early onset Alzheimer's disease and developing effective prevention and treatment strategies, we can continue to make significant progress in enhancing the quality of life for individuals impacted by this debilitating condition. And here are the references that we use for our project.